So now we will discuss more about the cell cycle, means what is the life cycle of the cell and how that cell division will be taking place. So cell cycle, if we want to define cell cycle, it is nothing but the sequential event where the cell duplicates each and every organelle, its majority of its genome, and synthesize the necessary all other constituents, and then eventually divide into the two equally or identical daughter cells. This is how we can define the cell cycle or cell division. So, if we want to divide them in the particular phases, there are four phases which play important role. First is the interphase. Interphase is nothing but the preparatory phase where the DNA duplication will be taking place and all other necessary proteins will be synthesized. Then there is a process of mitosis. Mitosis is nothing but the where the genomic separation or the chromosome separation will be happening, and we will get the two sets of chromosome separated. And in the third phase, that is the cytokinesis, where the cytoplasmic content will be divided equally into the two daughter cells and two new daughter cells will be formed. And there is one more phase which is a resting phase, that is what we call G0, because it is a non-dividing quiescent stage, means there is a no division, cell will be active, it's live, but it is a pause where whenever the cell division will be not happening at that particular phase. So let's see each and every phase in detail. So firstly, interphase. So interphase is the divided into the three parts, that is G1, S and G2. So G stands for gap. So gap is the one, means it's the phase where the synthesis of all the necessary parts will be like the proteins and the initiation signals will be given to the cell for the preparation for the cell division. This is the most lengthy phase in the whole cell cycle, which occupies almost 95% of the cell cycle's duration. So if we see this everything in phase wise, so first gap one is the first interval, means first mitosis cycle has already completed, and the uh, next cycle for the DNA replication will be start. So here this is the interval where this thing will be happening. So this is metabolically active phase, and cell will be keep on continuously growing in this particular phase. But no DNA replication will be happening into this particular duration. Then the next phase is a synthesis phase, that is S phase. So DNA synthesis and replication means firstly the DNA should be doubled, because that way it can be equally distributed in the newly formed cell. So the amount of DNA will be doubled. Uh, that way it can be evenly distributed at the end of the cytokinesis. So this is the only DNA synthesis phase. And then there will be the gap 2, which will be represented as G2, and this is the synthesis phase again, where the cell will keep on continuously growing and the synthesis of the proteins will be happening. Similarly, centrosomes division, means the division of the centrosome will be also happening into the interphase itself. So that those are the main region where they will be moving towards the pole and the spindle fibers will be formed at that particular point. And G0 phase, as we have discussed earlier, this is a quiescent phase. So when we say quiescent, that means the cell is not dividing, it is not inactive, it is in active mode. So it is inactive in the sense of it is not dividing in that way. So it keep on, it's not proliferating at all, but it is metabolically very much active and alive. So there are some cells in our body which are in this particular G0 phase. So one of the classic example is the heart cell of the adult animals, where it is metabolically active, but they are not dividing further once they get matured in the after a particular age. Then M phase, yes, yes, Shri.
Ma'am, the G1 phase
Hmm.
is like occurs between mitosis and the next phase, right? Like
The
it is
synthesis phase.
between
Synthesis phase.
DNA replication and mitosis.
Yes, yes, right.
And man, so but mitosis occurs after G1, right? After the interphase.
In the mitosis, well, first here you have to this is a circular event, right? So just consider this as a circle. So first mitosis is already done. So this phase is already finished. And then before starting with next DNA replication, this phase is in between. So if you want to see it in a sequential way, we see the interphase first, that is the preparatory phase, and then mitosis. We are thinking that it is a gap between the one mitosis is already occurred. The next phase will be starting. So this is in between the synthesis of the DNA and the first mitosis which is already done in that sense. So it's a circular event.
Okay, ma'am.
Okay, got it? So, mitosis is nothing but the M phase. So, mitosis is what we call, it is happening in the all body cells that we call the somatic cells in the scientific terminology. So, this involves the what we call the equational division. That is what we call it is, and it is restricted with to the diploid cells. So, we have already come across this terminology earlier also. Diploid is which have the two sets of the chromosomes. That is what we call diploid. And if I have only one set of chromosome, that is what we call haploid. So mitosis is characteristic of all the body cells which have diploid cell number in them. And this is the equational cell division. When we say equational, that means the chromosome numbers which are present in the parental cell will be have exactly same that of the daughter cell. So they have the similar kind of a genetic makeup in terms of the DNA constituents in it. So that way we are saying it is an equational division because there is no restriction of the chromosome. Means diploid cell, once it will be divided into two new daughter cells, the two newly formed daughter cells will be also diploid in nature. That way we called it as an equational division. So mainly the main characteristic that usually these cells are identical with that of the parental cells because the DNA is replicated and equally divided into two. So that way we say they are usually the identical in nature. So the main function of the mitosis means here we are talking about the division, so it is associated with the growth of the particular organism. The cells will divide and eventually the organism will grow. That is the very much main function of the cell division. And secondly, it is also play part in the cell repair. Means there are some injured tissues or injured cells will be there. Then cell replication also replace such torn out or injured or even killed cells, and that way the system can be get repaired. So the mitosis phase is divided into two broad things. First set we call the karyokinesis, and another second part is called the cytokinesis. So when we say karyokinesis, word karyon is associated with the nucleus. So nuclear division is what we call the karyokinesis. So karyokinesis phase will be divided into four stages: prophase, metaphase, anaphase, and telophase. So let's see what happened in each phase in detail. So first phase of the mitosis is the prophase. So prophase, what happened, as we are aware that the new means cellular system has all other organelles and everything, and as these are very long threads of the chromosomes that we have already seen, it's almost, if we constitute each and every part, there are 23 pairs of chromosomes, that is a 46 chromosomes, and they are very much tangled in nature. As they are linear, but they are very much tangled in nature. What happened in prophase? Here the condensation of the chromosomes will started. So that way we can visibly see the compact nature of the chromosomes is what is the main characteristic, or the condensation of the chromosome is the main characteristic of the prophase. Secondly, the nuclear membrane, which is another characteristic of the eukaryotic cell, here it is started to disappear. Then other organelles which are present in our body, in our body cells, they are also start to disappear. So here what happened, when we say the chromosome will start to condense, the chromosomes will see composed of the two chromatids which are held together by the centromere. So this is the basic structure that we are referring to, which is very much start to visible in the prophase itself. Then the duplicated centrosome, which phenomenon is already happened in the interphase, means the duplication of the centrosome happen in the interphase itself, which is started. This duplicated cell centrosomes will start separating and they are start to move towards poles. So when we say poles, these are nothing but the two ends of the cells. If we consider, so we have to consider for the sake of understanding, these are the poles of the cells. So these are duplicated centrosomes. So they are starting to move towards the each region. Then there are microtubules which are get radiated from the centrosome. When we talked about the structure of the centrosome in the last lecture, we have seen that there are radially arrangement of the microtubules are there. So these microtubules we call that astrals. So these astrals and the spindle fibers will start to radiate it from these centrosomes. Along with the astrals and the spindle fibers, we call this as a mitotic apparatus because they will start to pull the chromosomes and separate it together, which will be happening at the stage of the metaphase. So next phase is the metaphase. Metaphase, here we can see the complete disintegration of the nuclear envelope. Now there is no more nuclear envelope. So we can see the, yes, yes Shrey?
Ma'am, what are chromatids?
Chromatids are the two fibers that we have seen, that the two, each chromosome is composed of the two chromatids. So these are the two fibers which are get uh, means join or hold together by the centrosome. You are not present in the last lecture, right, Shrey?
Ma'am, your
Yes.
voice is breaking.
Am I audible? Am I audible now?
Yes, ma'am.
Okay. Have you gone through the last lecture's notes that I have uploaded already?
Ma'am, I was outstation and I came just at 10:30 today, so
Oh,
I did not see.
okay, okay. So actually this part we have already covered, the structure of the chromosomes. So chromosomes are the nothing but it is made up of the two chromatids. So these are the, we can say these are the chromatid fibers which are held together by one central disc-like portion which is called the centromere. Okay, so chromatids is on each chromatid we have the DNA in it. Okay, so the it is a threads of the DNA which are held together, two threads of the DNA which are held together by the central proteinaceous structure, that is what we call the centromere. And on the centromeres inside the main center of the centromere, we call it as a kinetochore. Okay, is this part clear?
Okay.
Okay, so in the metaphase, so the condensation of the chromosome will start in the prophase itself. So in metaphase, the nuclear envelope is completely disappeared. Now the and all our remaining organelles are also disappeared in nature. So only cytoplasm and our chromosomes and the remaining mitotic apparatus structure will be there. So metaphase chromosomes are two sister chromatids which are held together by the centromere. That is the basic structure that we can say. And on the small disc-like structure which is present at the surface of the centromere is what we called as a kinetochore. So this is a site, on the centromere there is a kinetochore which is the attachment site of the spindle fibers. So the spindle fibers which are present from the centrosome. So please don't get confused between centrosome and centromere. Centrosome is the one which is the polar structure which is developed at the cell divisional cycle. So you can consider this as a central point where the fibers are attached, and centromere is nothing but it is a disc-like portion which are holding together two chromatids, and on the center or on the surface of the centromere there is a disc-like, small disc-like structure. One more structure is there which we referred as a kinetochore, which is the attachment point for these spindle fibers. So there is a metaphase plate-like structure is formed. What is the plate-like structure? This is nothing but the plane alignment of the chromosomes. So all the chromosomes will align in a particular plane in a straight way, and then these fibers will get attached to each center. This is how the structure will be developing. And this whole structure, we refer it as a mitotic apparatus. So once the attachment will be formed on this particular region, the chromosomes will move towards the spindle equator. This is how the each pole will play important role to attract each chromosome on its side for the equal division. So in the metaphase, the metaphasic plate will be developed. Spindle fibers get attached to the kinetochore of each chromosome, and everything will be prepared to pull out in the next phase. Then in anaphase, what happened? All the spindle fibers will get contracted towards the centrosome. So in this way, chromatids will move towards the opposite poles. Now here the chromatids and each centromere will be get pulled towards the opposite pole. So each chromatid will have its own centromere, and as the DNA is already duplicated, each set will have the equal quantity of the DNA in its, because it's already replicated in its phase of the interphase. And after once they pull towards the opposite poles to each other, then at the end of the telophase, which is the last phase, already the chromosomes is already chromatids is already separated, so they are starting to cluster again. So now they lose their identity being a condensed form. They are more discrete elements because now they are starting to rewinding again, and at this stage the nuclear envelope will start to reform at their poles. Also, nucleolus will be formed, reformed. We can say more or formed. It will be reformed. Golgi complex, ER, the other endoplasmic reticulum, all other organelles start to reform at this particular stage. But still, the two things will be present in a single cell. Cells are not still divided into the two cells. This is the basic phases of the mitosis. Or the or we can say karyokinesis completes at the end of the telophase. So now everything is duplicated. So now need to prepare the two new daughter cells, which will be happening at the end of the cytokinesis. So cytokinesis is nothing but a division of the two daughter cells and by separating their cytoplasmic content itself. So by this way, the different organelles which are present in the cell, apart from the chromosomes, like the chromatids plus the mitochondria, the endoplasmic reticulum, and everything will be distributed into two daughter cells. This is what happened at the end of the cytokinesis. So, there is a difference between the animal cell and plant cell, how the cytokinesis will be taking place. In case of the animal cell, simply there is a furrow-like or the notch-like region will be started to form between in the plasma membrane itself, which will be gradually gets deepened, and then the two cells will be separated. So this is how the furrow or the notch-like projections will be formed, and two daughter cells will be separated in this particular fashion for the animal cell, because here the only the plasma membrane notch will be enough, because animal cells do not have any cell wall. On the other hand, plant cell already have the cell wall in it. So for this particular, wall formation should be there. So here the wall formation will be start. So wall formation from the center of the cells, and it start to grow outward, and by this way the new cell wall will be formed. So here cell plate formation will be there in case of the plant cell. And once the cell plate will be formed, it is the it is nothing but a middle labella which will be joining the two adjacent cells. So when we talked about the cell structure in case of the cell wall structure in the earlier chapter, we see there is a primary cell wall, secondary cell wall, and there is a middle lamellae. So middle lamellae is nothing but this is the adjacent portion between the two cells once the wall is the developed, which is developed once the cell divisional part is completed for the plant cell. So here in plant cell, the cell plate will be formed by the wall formation, and in case of the animal cell, a notch-like projection, that is what we call the furrow, is developed, and which will eventually divide the cytoplasmic content, and the two new daughter cells will be formed. In some cases, yes, Shrey.
Ma'am, what are lateral walls? Lateral means what, ma'am?
Lateral means in the cell wall structure, what happened? So, cell wall structure, just imagine this is a cell wall structure of the plant cell. So, there is an adjacent cell in between. So, these are the lateral walls which are present. The divided cell will be present to the next cell, right? So, the wall formation is nothing but the lateral wall which will be developed by the presence of the middle lamellae, which is adjacent region or newly formed wall in between.
Okay, ma'am, got it.
Okay. So by cytokinesis, we get two newly formed daughter cells from the parental cell itself. However, in case of the some specific cases, what happens? The karyokinesis is complete. That means the telophase will be achieved. However, it is not followed by the cytokinesis. That means the two separate means the nuclear division is already completed, but the new cytoplasmic content is not differentiated. So this will result into the multinucleated conditions because the nuclear division or the karyokinesis is already complete, the cell division part does not happen. So this condition where the karyokinesis is not followed by cytokinesis and which results into the multinucleated condition, this phenomenon we called as a syncytium. So this syncytium is a characteristic of the liquid endosperm of the coconut. Is nothing but the tender coconut water that we drink. Is the nothing but the liquid endosperm of the coconut, which is the classic example of this syncytium, where the divisional part will be taking place, but the cytokinesis don't happen. So is this part clear? Mitosis part?
Yes.
Okay.
Yes, ma'am.
Okay. So this is the main classic or basic idea of cell division is nothing but about the mitosis. Now the second type of cell division is called meiosis. So meiosis is a special kind of a cell division, is what we called as a reductional cell division. Why reductional cell division? Here the chromosome number will be in the next newly daughter form or daughter cells that will be developed will be haploid in nature. That means the daughter cells will get only one set of the chromosomes. That is why this is a reduction in from the diploid nature. So that's why it's the meiosis is known as a reductional cell division. However, the mitosis what we call the equational cell division. That is the basic difference. So this is a special kind of a reaction that is happened where the two times the nuclear division will be taking place or the cell division will be happening two times. However, the DNA replication happens only one time. That's how we can get the haploid progeny at the end. So the mitosis or the process of the mitosis have only one cycle. That is the M phase will be divided. The karyokinesis will happen only once. However, in case of the meiosis, it happens two times. So the cell division part is divided as meiosis 1 and meiosis 2. So meiosis 1 is the initial phase of the where the parental chromosomes will be replicated. So in before happening meiosis 1, the interphase will be happening. Interphase will followed by meiosis 1, and before meiosis 2 no interphase will be happening. The immediately meiosis 2 will be start. So this is how the sequence will be there. So here the word that we are using is the homologous chromosomes. So we have used the words chromatids. So chromatids is nothing but the two threads of the DNA, more in simpler language, is held together by one centromere. Here we are talking about homologous chromosomes. As you are aware, in each cell we get the two sets of the chromosomes. We have 23 pairs of the chromosomes. That means each one we get one from our maternal side, that is one from our mother and other from our father. That is how we get the two sets of the chromosome. So this is what we call the homologous chromosomes. So here what happens? Here firstly the chromosomes will get separated and then chromatids will get separated. How? And here another important characteristic that the mitosis have that we have the identical sets of the genetic material because the things will be happening, there is a replication, so equal sets of the DNA will be prepared. However, the meiosis is characterized by there is a recombination event happen. That way, the new combination of the genetic material will be prepared, and that's why we say the progeny is different from that of the parental cell. How this is happening? So this is quite lengthy phase. So meiosis 1, the phases will be same in the karyokinesis, prophase, metaphase, anaphase, telophase. For the sake of understanding, the meiosis 1, we called it as a prophase 1, prophase 2, like so. Meiosis 1, prophase 1 is quite extensive and it is quite lengthy phase. Why? Here all the recombination event will be happening. So prophase 1 is again divided into 5 other stages. So first stage is called the leptotin. So here the nuclear envelope in the cell will be still there. The chromosomes will start to condense as that of the what happened in the prophase. So here for the sake of understanding, you can see the two chromosomes will made up of the chromatids, right? So here blue will be just imagine for the sake of understanding, I'm there. Blue set is get from the set from the father and red is from the mother. So each have the two chromatids in it, so which will be represented by dark and faint colors in it. So here the firstly condensation of the chromosomes will be start in the stage of the leptotin. Then the phase of the zygotin happens. So what happens in zygotin? The chromosomes will start to pair with each other and synapse will be formed. So when we say synapse, synapse is nothing with nothing but the two chromatids will come together. That is a non-homologous chromosomes. Means one set of the chromosomes. This is one set. This is from father and another is from mother. So this is from mother. So when I refer this, these are the sister chromatids because they are related with each other. However, the chromatids which are not related with each other means one from the father and one from the mother, we will refer it as a non-sister chromatid. So between this non-sister chromatid, because of the close proximity that they have, they will start to form the synaptonemal complex. That way they are coming close proximity to each other, the non-sister chromatids, which is characteristic of the zygotin stage. Then the stage of patchitin happen. So just consider this is a patch formation between the non-sister chromatids. So what happened? The chromosomes are fully synapsed and the crossing over will be start. So here you can see a knot like projection is happening in this particular phase. So two non-sister chromatids, that is the one chromatid from the father and one chromatid will from the mother will be start to cross over on each other, and that way a complex will start to form. A patch formation will be starting. Then the next stage is the diplotin. So in the diplotin, the chiasma formation will be there. Chiasma formation means after the synaptomemal formation, the chiasma will be there. So that way, here you can see in the non-sister chromatid of the father, the small portion from the mother is get attached, and on the non-sister chromatid of the mother, the small blue portion is get attached. This is what we call chiasma formation, and this is the source of the recombination event. That way, the chromosomes will have different combination of the genetic material. That's why the characteristic of the children is different from the parents in some cases, because some things are identical. However, because of the new combinations which will be developed in this prophase recombination phase, the new combination or the new genetic makeup that we have for the progeny cells. And after diplotin, the last stage of the prophase 1 will be called the dikinesis. Is nothing but this is the for once the sharing will be done, the further chromosome will start to contract, means they will start condense. Here the sharing or the recombination event is already completed. So this is quite extensive phase, and this is one of the important phases which will be more time asked as per your exam point of view. So please do remember this diagram very clearly, that where the synapse will be happening, where the chiasma will be happening, and how the separation will be happening at what stage. Is this phase clear? Any doubts? Yes. Yes, Shri.
Will the chiasma be there in dikinesis?
Already the separation will be there. So here you can see the red non-homologous chromatid already get the part of this particular. So chiasma is already completed in diplotin. And further the once now they are consider this as a one chromosome, and this will be the another newly formed, newly recombinant chromosome. So chiasma will not be present in dikinesis. It is present in case of the diplotin.
Okay.
Okay. So this is most extensive phase and which will take the most time for the meiosis phase. So after prophase 1, then will be the metaphase 1. So the characteristic that we have in each phase will be remain same. So the metaphase plate will be formed same as the mitosis, and the centrosome will be there. The kinetochore will get attached with the centrosome spindle fibers. The every characteristic is the same. Now, the difference that we have to remember about meiosis once metaphase or anaphase, here the pair of the homologous chromosomes is we are talking about. So here homologous chromosomes will be aligned at the equator, and in the anaphase 1, the homologous chromosomes will move towards the opposite poles. Here we are not talking about chromatids, here we are talking about chromosomes. So chromatids is nothing but the single fiber we are talking about. However, just say here we are talking about the whole chromosome itself. So 46 chromosomes will be there at the time of the metaphase 1. However, when anaphase will be happening, the pairs will be get separated, and we can say that in the anaphase at each pole 23 chromosomes will get pulled out. So even the recombination event which will be happen which has already happened in prophase 1, here in the diagram you can see there are new patches which will be resulted in the prophase 1 is represented by different colors. So the new combination of the genetic material is already prepared in each set of the chromosomes, and then they are getting pulled towards the opposite pole, which will be happening in case of the anaphase 1. So after anaphase 1, telophase 1 will be happening where the nuclear envelope will be reformed. Two new cells will be formed which are haploid in nature. That is very much important. So it has the only one set of the chromosome in each. So new nuclear membrane will be formed and the cytoplasm will be started to divide. So at the end of the meiosis 1, two newly formed daughter cells will be there which are haploid in nature. That is, it contains only one set of the chromosomes. So if we want to summarize this whole thing in one diagram for the sake of better understanding, what happened in prophase 1? So prophase 1, the sister chromatids will be there, and the homologous chromosomes, that is one set from the father and another set from the mother, the non-homologous chromatids will get crossing over. Then it will be result into the new recombination event will be happening by the chiasma formation, and ultimately the newly rearranged or recombined chromosomes will condense at the end of prophase 1, and nuclear envelope will start to disappear. In metaphase 1, nuclear envelope will be completely disappear. Metaphase plate will be formed. That is, the chromosomes will be arranged in a plane, and the centromere will with the kinetochore will get attached with the spindle fibers which are present at the centrosome. Then in anaphase 1, sister chromatids remain attached. That is, the only homologous chromosomes will get separated at each region. And at the end of the telophase 1, newly formed daughter cells will have only one set of the chromosomes. So say, for example, in case of the humans, we have 46 set of the chromosomes. So at the end of the meiosis 1, each newly formed daughter cell, that is the two cells that will be formed, only have the 23 chromosomes in each. Is this part clear?
Yes, ma'am.
Okay.
Yes, ma'am.
So after meiosis 1, meiosis 2 will be happening. So when we say meiosis 2 is exactly same as the mitosis. So the phases will be same and the understanding should be same. So what happened at the mitosis? At the end of the mitosis, whatever chromosome number that will have in the parental cell will be have exactly same as the have the same in the daughter cells. So that means if we have the parental progeny as haploid in nature, then the daughter cells will be resultant after meiosis 2 will also have the haploid in nature. So what happens in meiosis 2? In prophase 2, the nuclear membrane will start to disappear. It forms the compact condensed chromosomes again. And in metaphase 2, the chromosomes will align in the metaphase plate and the spindle fibers will get attached. In case of anaphase 2, what happens here? The chromatid separation will be happening. Here the chromosomes no. Here we are talking about the mitosis. Imagine the thing same what happened in the mitosis. So here chromatid separation will be happening because DNA is already duplicated before meiosis 1. So each will have already a very huge chunk of DNA in it which is already duplicated. So each chromatid will get equal set of the chromosomes which is already there. But however, here the recombination event is already happened. So you can see the patches of the different colors over here which represent at the recombination event which is happening in each chromatid. So here in the anaphase 2, chromatid separation will be happening at each pole of the cell, and at the end of telophase 2, two newly formed daughter cells will be there. So here the starting material for the meiosis 1 is two parental cells which are result of the meiosis 1. So at the end of the meiosis 2, total 4 haploid progenies we get, or the 4 haploid daughter cells will be getting, which have the very different genetic makeup than that of the parental cell itself. So here we call the formation of the tetrad. So word tetrad is associated with the 4 cells. So these are the 4 haploid cells which are formed at the end of the meiosis 2. Any questions?
Ma'am,
Hmm.
ma'am, which cells perform meiosis?
Meiosis 2 is performed in the sexual cells. So when we say the formation of the sperms or formation of the egg in case of the human cells that we can refer to, only those special sex cells, this kind of a division will be happening. Because ultimately the egg and the sperm will reunite to form the zygote, and that will be eventually develop into a new human being. So two haploid cells when unite together, they will get each 23 sets of chromosome with them, and which will form eventually a diploid organism which again have the 46 chromosome in it. So meiosis 2 is happen in case of only sex cells.
So sex
Hmm.
cells are germ cells, right?
Yes, sex cells are germ cells, right?
Ma'am, then gamete, what are gametes?
Gametes are the means the egg or the sperm is what we called as a gamete.
And ma'am,
Hmm.
gamete and gametophyte are the same thing, right?
Gametophyte is a terminology that is associated with the sexual reproduction in case of the, especially for the plants. So we use the sporophyte and gametophyte. So spore formation is usually happen by the asexual way, and gametophytes where the two gametes get united to form the sexual way in case of the plants. That is what we call the gametophyte. But the conceptual understanding, it is associated with the sexual way. That is correct.
Okay, ma'am.
Okay. So let's summarize this because the sequentially by the understanding the events, it's very easy. You have to just remember what is the difference between the phases and what are the different characteristic it is associated with. So let's see, let's compare the meiosis 1 with meiosis 2. So meiosis 1 is a heterotypic division and meiosis 2 is homotypic division. So homo is the word associated with the similar when we say, and meiosis 1 is what we call hetero is a different and homo is similar. So because the heterotypic division means what we want to say, meiosis 1 involves the formation or the recombination event is there. The parental progeny will be different from the daughter progeny. That way it is heterotypic in nature because the reduction will be happening in the genetic material. On the other hand, meiosis 2, the it is similar like a mitosis. That is what happened in the mitosis. So it is again equational kind of a division that is happening. That way it is referred as a homotypic division. So in the meiosis 1, the reduction of the chromosome number will be happen in case of the daughter cells. On the other hand, in case of meiosis 2, it equalizes the number. So it is the equational division. So both parental and daughter cells have the same number of the chromosomes in it. It is not reduced further. Then in case of the meiosis 1, it is the homologous chromosomes which are present in the beginning. So means one set from the father and other from the mother. So in the beginning material will be the homologous the pairs of the homologous chromosome will be there in case of the meiosis 1. In case of meiosis 2, individual bivalent chromosomes will be there because only one set of the only one chromosome will be present from the each set. So it's an individual bivalent chromosome. So the chromatid separation will be happening at the end of the meiosis 2. So the phase wise, for the sake of understanding, we divided into prophase 1, metaphase 1, anaphase 1, telophase 1. On the other hand, meiosis 2, we divided as prophase 2, metaphase 2, anaphase 2, and telophase 2. In case of the meiosis, individual chromosomes are present in each daughter nuclei. So here we are talking about the whole chromosome itself, which are bivalent in nature, means two chromatids will get attached to it. On the other hand, meiosis 2 will have the sister chromosomes which are present in daughter nuclei. So the chromatids will be there. When we say sister, word sister is a related one. That way we called it as a sister. And on the other hand, in case of the meiosis 2, two sets will be already present in the individual way for the meiosis 1. In case of the meiosis 1, chromosomes are crossing over. That is happening in case of prophase 1, which is the event where the recombination will be taking place. On the other hand, in the meiosis 2, no crossing over will be happening. It is just whatever the chromatid composition will be there, they will be divided based, they will be just get separated based on that. As it involves various other events like the chiasma formation, synaptonemal complex, we can easily say that meiosis 1 is the more complex division and which will take more time than what happened in case of the meiosis 2. And the main characteristic that meiosis 1 is preceded by interphase. So interphase will be happening only one time before meiosis 1. After meiosis 1, there is no interphase again happening before meiosis 2. After meiosis 1, immediately meiosis 2 will be starting. So no interphase will be taking place before meiosis 1, meiosis 2. So if we want to make means what is the similarity between the two different kinds of a cell division, that is mitosis and meiosis. Firstly, see what are the similarities in between them. Firstly, they occurs in both plant and animal cell. That is the main thing. It starts with the diploid parental cell. So first initial material for the both kind of a cell is a diploid cell, which will eventually produces haploid progeny or haploid daughter cells in case of the meiosis. It will remain diploid in case of the mitosis. The main purpose of the cell division is the production of the new cells, which is the common in case of the both mitosis and meiosis. Cell undergo DNA replication before mitosis or meiosis or actual cell division will be taking place. And the basic stage, that is the prophase, metaphase, anaphase, telophase will be happening, means the phase wise they are happening in the same way. That is the common point in between mitosis and meiosis. If you want to say the difference what they have, the mitosis occurs in all organisms, means except viruses, of course. They are happening in all kinds of organisms. Meiosis occurs only in case of plant, animals and fungi, where the gamete formation or the sexual reproduction, the one we have which have the sexual reproduction ability, this happens only in those organisms. Mitosis creates all body or the somatic cells that we call. All kinds of cells are the body cells are produced by the process of the mitosis. On the other hand, only sex cells or the germ cells like sperm or egg is produced by the process of the meiosis. Mitosis involves only one cycle of the division, means only once the karyokinesis will be taking place and then cytokinesis will be happening. On the other hand, meiosis involves two cycles of the cell division. So firstly, meiosis 1 will be happening and then meiosis 2 will be happening. Then in case of the mitosis, which results into the uh, diploid daughter cells which will be produced and which are identical with that of the parental cells. On the other hand, at the end of the meiosis, the four haploid daughter cells will be produced, which are genetically different because of the recombination event also, and because of the genetic makeup, means the parent will have the diploid in nature. On the other hand, the daughter will have the haploid in nature. So genetically also they are different, and the number wise also the daughter cells produced in the meiosis are different from the parental cell. So in case of the humans, we can see it created 46 chromosomes in case of, because all the body cells have 46 chromosomes in it. On the other hand, meiosis 2, at the end product will have, the daughter cells will have only 23 sets of the 23 chromosomes. That is, only one set of the chromosome is present in case of the daughter cells of the meiosis. So I have just summarized everything. We have seen all the phases in at different level. So just for the sake of the understanding what is the characteristic of the each phase, I summarize this particular part. So these are some electron microscopic things which we can see more clearly what happened actually at the cellular level than the just comparing at the diagrammatic level. So these are the, so even if you can see this chart in together, you can easily remember what happens at each phase. So in prophase, the condensation of the chromosome will be happening, nuclear envelopes start to disappear, and the spindles will be re-emerged, centrosomes will start to separate. In pro-metaphase means the metaphase will be divided into early and late, the way we can say. So in the early metaphase, what happens? The chromosomes condense itself, the kinetochore will start to appear on the centromere, spindle fibers will start to move towards the opposite pole, means it start to attachment, and the metaphase plate formation will be happening in the metaphase, or we can say it as a late metaphase moreover. Then in anaphase, it will pull down the spindle fibers will pull down each sister chromatids, and they will now termed as a new chromosomes, and they will move towards the opposite poles. In the telophase, the nuclear membrane will start to reappear. The condensed form of the chromosomes will gone, and they will start to moreover dense in nature, and the other uh, kind of organelles like endoplasmic reticulum, Golgi apparatus will start to reappear at this phase. And at the end of the cytokinesis, the cytoplasmic content will be divided into two parts, and then ultimately newly formed daughter cells will be there, which will be have identical genetic makeup that of the parental cell. In case of the meiosis 2, this is summary of the meiosis 2, which is happening in case of the germ cells. So word gonad is associated with the sex organ. So in case of the male, the sperms are produced in testis, and in case of the female, the eggs are produced in ovaries. So at that time, we referred ovaries and testes as gonads, and the germ cells are responsible for producing this particular stage. So in the first stage, the interphase will be happening. That is the duplication of the DNA will be, replication of the DNA will be taking place. In case of the meiosis 1, the prophase 1 is the most characterized one, where the synaptonomial complex will ultimately form the chiasma, which makes the recombination event between the non-sister chromatids. And here what happens? The segregation of the homologous pairs of the chromosomes will be happening, and the chromosome will be number will be divided into half at the end of the telophase 1. On the other hand, meiosis 2 will be immediately happen considering the end product of the meiosis 1 as the starting material, and this is the equational division. Here the sister chromatids will get separated, which produces the 4 haploid gametes at the end of the meiosis 2, which are genetically different and also contains the half of the number of the chromosomes than what is the present at the starting material for the meiosis. Is this part understood? Conceptually
Ma'am,
clear?
indeed.
Yeah. Yes. Sorry, Shrey, you are not audible.
Hello, ma'am. Hello.
Yeah, yeah, yes. Shrey, sorry, I can't hear you.
Uh, Hello, ma'am. Hello.
Yeah, yeah, yeah. Now you're audible. Yes. Uh,
Assortment.
So I guess I can't hear you clearly, but I think you are asking about independent assortment, right?
Yes, ma'am.
Oh,
Yes.
yeah. So the word independent assortment is associated with the separation event, means all the characteristic expressed individually in each generation. So this particular word, segregation and independent assortment, these two concepts we come across when we talk about the Mendelian separation. So word independent assortment. So here two sets of chromosomes will get separated. So each cell will get its own characteristic from the only one chromosome which will be present in each in at the end of the telophase one. So anaphase 1 is associated with the independent assortment, that is the separation of the different characteristics, and each newly formed cell will get only one of its kind. So that is how it is independently expressed and independently gets separated. That way we use the terminology independent assortment. Any other question?
Ma'am, your voice is cracking in the middle.
Oh,
After
okay,
2 or 3 seconds.
okay. Okay. It's still unclear.
No, ma'am, but
in
sometimes.
between in between happening, but I hope you are able to hear things or understand things clearly.
Yes, ma'am.
Okay, great. So that is what about the cell division and cell cycle. So let's solve few questions based on this. Hope you have understood the concept very clearly. So Shrey, if you are unable, means if I am unable to hear your voice, you can type in the chat box your option, no problem at all. So here is
Okay,
your
ma'am.
okay. Now I can hear you, but in between I don't know. I am also unable to hear you. So here is your first question.
Ma'am, option A.
Option A. Okay. Danish, what about you?
No, ma'am, confused between A and C.
A and C. Okay. So definitely we can rule out B and D because here we are talking about mitosis, so no crossing over events will be there. So we can eliminate both things. So here option A is the condensation, which will be happening in case of the prophase. So nuclear membrane disassembly will be happening at the early itself in the metaphase. Arrangement at the equator, which is a characteristic of the metaphase. Then centromere division, which is once the chromatids will be get separated, each uh, set will get its own centromere. So that way the centromere separation will be there. Segregations means the two sets will be separated completely, and the telophase will be happening, which is the absolutely correct. Option C is the condensation, arrangement at the equator, centromere division, segregation, and telophase. Though phase wise, means step wise, it is correct. Option A is the more correct in the way here the few more stages, that is the nuclear membrane disassembly is also mentioned, which is happening after condensation. So this is the most appropriate one. So option A will be the correct one. Next.
Ma'am, is it option B, G2M?
Option B, okay. How about you, Shrey?
Ma'am, option A.
Option A, okay. So we are talking about this is what we are saying, the DNA replication fork is stalled. So that means it has stopped somehow. That means DNA replication fork means DNA synthesis has already stopped at some point once the fork is formed. So that means S phase is already initiated. So if S phase is already initiated, that means G1 phase is already done. So G1 phase is already done and things are not happening at the S phase itself, which has already started. Okay, so if this is the case, then predominantly active phase will be G2M, which will be followed after synthesis phase, which will decide whether to start with G2 or M, or the checkpoints will be work at the G2 and M, because S phase is already start and G1 phase is already finished. So the next phase will be work as a checkpoint, whether it should be entering into the mitosis or not. So option B will be the correct one. G2 and M will be predominantly activated as a checkpoint.
Ma'am, what does stalled DNA replication fork mean?
Stalled means stop. DNA replication. Have you learned about this? How DNA replication will be happening?
Yes, ma'am, but
Yes.
when X phase is completed and G2 phase has started, isn't
Huh?
the DNA replication completed?
Yes, the DNA replication is completed. That is true. But here the question that we have asked, when the cell has stalled DNA replication, that means DNA replication fork is formed, but it is not replicated further somehow. Means some kind of error is there. We can imagine like this way. That means cell has already started working on the S phase. G1 is already completed, and the checkpoint is for, means in the cellular way, they will check the G1 is completed, DNA replication is completed, check. Then after that, G2 will be started, and G2 and M means whether to the whole process is preparatory material is there to start the mitosis process. Is there is at each level there is a checkpoint. So if the cell is stalled at the DNA replication fork, that means the S phase is already started. G1 phase is completed. Unless G1 phase completed, the cell won't enter into the synthesis phase. So G1 is already done. S is already started, but it has stopped somehow. The DNA replication fork is not working somehow. So that means the predominantly active phase for the next checkpoint, that cell cannot be entered into the M or G2 or M phase. That means the G2 or M will be the next checkpoint. That way, cell won't enter into the mitosis.
So, ma'am, is that
Hmm.
the quiescent phase?
No, we cannot say it's a quiescent phase, but yeah, we can. It's moreover the kind of the error which we can say it's a quiescent. It's a kind of a inactivated phase, but not dividing phase. Am I audible?
Yes, ma'am, just for
Okay.
2 3 seconds.
Okay. So that's what I'm saying. Though we can say it's a quiescent phase in the form of the inactivated, because the cell won't be dividing further for unless the error will be repaired some or other way. But this is just for the sake of the case understanding that we are talking about, that which checkpoint will be worked on this particular thing or not. But yes, we can say this cell will be will not be dividing further, but it is still metabolically active. So it is a kind of a quiescent phase, but we cannot say it's a G0 phase, because G0 phase will be happening after one mitosis sample, mitosis process is already done. For some or other reason, whether to enter into a G1 phase or not is the checkpoint. But between mitosis and G1, the G0 phase will be there, where the cell will be further decide to enter into the interphase itself or not. So this kind of a middle stage will be the quiescent stage, or some kind of a repair mechanism will be required to reinitiate the synthesis for the cell. So the next checkpoints will work that yes, the next we cannot go further for the mitosis phase itself.
Ma'am, so will
Hmm.
G2 happen as there is a
No,
stage
no,
S
G2
phase?
won't, in such cases, G2 won't happen. The checkpoints or the system will say there is an error in between, so G2 won't be happening at that time. So there are some repair mechanisms in our body which will work, or otherwise some other cellular mechanism will be taking the call on that particular thing, that it is something seriously wrong with the cell or like so.
So from incomplete S phase, it will directly go to the checkpoint.
Yes, the checkpoint is nothing but just consider this as a checkpoint that we have on the road itself. That first phase is completely happened. So that is the safe, means we have already done this correctly. Now the cell will enter into the next phase. S phase, the complete DNA is doubled or not? Yes, it's completed. That is the checkpoint. So whether the once the completed, the required proteins will be synthesized or not, which will be again happening in G2, the required proteins will be synthesized. So unless the first phase itself is incomplete, how would the cell enter into the next phase? So G2 or M means that will be the stall point for the cell whether to enter into the mitosis or not. In that way, we are saying it as a checkpoint.
Ma'am, so there are only two checkpoints
In a way,
between
means
G1 and S and G2 and M.
yes. Complete the interphase itself.
Okay, ma'am. So there isn't
Okay.
a checkpoint between S and G2.
S and G2 means here for the sake of understanding it has been given whether to enter into the next phase or not for that way. But once one phase is completed, it is communicated that the next phase will be start. So checkpoint in that way, the because the protein synthesis or other required machinery will be happen, because here though we are thinking this as a sequential event, there are many things happening in the cell, right? So there are much more simultaneous coordination happen inside the cell itself. So for the sake of understanding, for this particular question, they have framed in this way. So for amongst the options that they have given, we can say whether uh, to enter the cell itself into the G2 or M, means in that between checkpoint should be there, means in for the both things or not. In that way, we have to understand this question.
Okay, ma'am. Understood.
Okay. Next.
Option D.
Core of-
Yes, kinetochore of the chromosome. Option D is the correct one.
Ma'am, what is kinetosome?
Kinetosome, no, they have just, it is just made up terminology, just to
Oh,
confuse.
ma'am, only G2.
Okay, only G2. How about Shrey?
Yes, ma'am. Option
G2?
C.
Yes, it's correct. It's actually a bit confusing question itself, we can say, because G2 will be the means G1, that cell will be remaining diploid because no DNA replication will be there. In S phase, the DNA will be doubled. So 4C is already achieved. In G2, the phase 4C will be remain as it is on complete phase of the G2. In case of the M, the initial quantity will be 4C, but at the end, it is
M
divided into the
phase.
M phase.
Sorry, ma'am, I couldn't hear you.
Okay, so in G2 phase, the amount is already double. That is 4C will be remain at the same level. So that means the only G2 is the correct answer. However, in case of the M phase, when the cell will enter into the M phase, at that time the 4C level is already there, right? Because the DNA is replicated. Then eventually at the end of the telophase, it is divided into two daughter cells and two new daughter cells will be formed. So for the here we are talking about the amount of DNA in a cell remains 4C level. So as the word remains is emphasized for this particular question, only G2 is the correct answer. Okay.
Ma'am,
Yes.
ma'am, what is like C? C.
See. See here the concentration word, you can use the word as a concentration. So when we usually put the word N, that is diploid, is represented by 2N. That means two sets of the chromosome. So n is represented as a number. Similarly, here they have represented by the C, nothing else. So the concentration means already deployed in nature, the DNA concentration is doubled than the initial concentration. So that way they have just represented the C, nothing else.
Okay, ma'am.
Next. Is my screen visible to you all?
Yes, ma'am.
Okay, okay. Yeah. It is just pause here for somehow I don't.
See,
Sorry, I can't hear you clearly. C, okay. How about you, Danish?
I'm confused between B and C.
B and C. Okay, so it's telophase for sure. That is agreed by both of you, which is good thing. Now the B option B is endoplasmic reticulum and nucleolus not reformed yet, and option C is nuclear envelope reform and Golgi complex reform. So if we see the at the end of the telophase, the main characteristic that we can define for the cellular environment, the nuclear envelope will be reformed, which is the main characteristic what happened at the end of the telophase. For the both ends, the nuclear envelope will be formed and the Golgi complex ER will be start to reappear. So they will start to reform. Similarly, endoplasmic reticulum nucleolus is also started to reform at the end of the telophase. So this is a incorrect option. This will be the correct explanation for this diagram. The nuclear envelope will be formed, which we can see very easily over here. In Golgi complexes, ER nucleolus will start to reappear at the end of telophase itself. Next.
Now I had a doubt.
Yeah.
Ma'am, since 1 or 2 is not mentioned, can we conclude that this is mitosis only?
Yeah, here you don't need to judge about whether it is mitosis or meiosis. Irrespective of that, the characteristic associated with all these phases are remain same, right? Don't consider here. You have to here just a cell divisional diagram. You don't have to worry about whether we are talking about this is mitosis or meiosis. The characteristic of the each phase, that is prophase, metaphase, anaphase, telophase, will remain same, right? The condensation so here just think about the general phases of the cell division irrespective of the mitosis or meiosis. So as per option, firstly read the question, and as per you have to think as per option point of view in the for such questions. Hmm? Next.
now option D.
Option D. Yes, option D is the correct one. The ER and other nuclear organelles will start to disappear in early prophase itself, and nuclear membrane will start to disappear it completely disappeared in the pro-metaphase, that is the early metaphase. The nuclear membrane will be completely disappeared. However, the other organelles start to disappear, begin to disappear in early prophase itself. Next.
And ma'am, they
Hmm?
completely disappear at early metaphase.
Which one? The other organelles?
Yes, ma'am.
They will completely disappear in late prophase itself.
Okay, ma'am.
Ma'am, could
So
you
only
repeat?
the nuclear
Nuclear
membrane.
membrane. Yes, nuclear membrane, because after the nuclear membrane will completely disappear, the things will be entered into the metaphase itself. The metaphase plate will be formed, right? After that. So it requires that space for the centrosomes and everything to form in this particular region.
So, ma'am, will these disappear before the nuclear membrane?
Yes, in the
Okay, ma'am.
late prophase itself, they will uh, disappear. Next.
Anaphase, option C.
Option C. Okay.
Option A.
Option A, metaphase. So now see, with the question that we have asked, we want to count the number of the chromosomes. Okay, this is the basic requirement is we have to count the number of chromosomes. If this is the case, they should be visibly very clear i- by the process of the staining or whatever the method that we are using. So if this is the case, prophase will be the just condensation phase. They are not still separated. We want the more the separation, we can easily see them clearly. In case of the metaphase, they are already all the chromosomes are arranged in the metaphasic plate itself, and they are in the condensed form. So that way they are much more visible and easy to count. In case of the anaphase, the chromatids will get separated, right? So the at that time the already two pairs will be means that they are already separated in two formats, and they are pulling towards the end region. So at that time the counting will be more difficult. They are more condensed and arranged in a specific line or specific plate, will be more visibly easy for us to count them. So that way metaphase is very easy for the counting. So option A will be the correct one. We want our requirement is counting of chromosomes.
Ma'am, could you please repeat question 7?
Okay. So we were talking about we want to our need for this question is we want to count the chromosomes. So by this way, we want to see them in more clear format. So in metaphase, all the chromosomes are in condensed form and they are arranged in a plane. So at that time, it is very easy for them to count uh, by method of the staining or anyways. In case of the anaphase, what happened? The chromatids will be get already get separated. So we get already get two sets of the means chromatid separation will be there. So two sets will be there. So visibly, it will be difficult for us to count the chromosomes more clearly because they are already pulled towards at the end. So the aggregation or the by the staining way also it will be very difficult. So the condensed and arranged in a particular fashion will be more easy for us to count. So that way metaphase will be more suitable for uh, counting the chromosome number. So option A will be the correct one. Next.
Ma'am, option C.
Yes, option C is the correct one because meiosis 2 is the equational division because it is same as the mitosis. What happened? The separation of the chromatids will be taking place, which is a characteristic of the meiosis 2. Next.
Ma'am,
Yes.
option A and option B are the same, right? Like crossing over and pairing of homologous chromosomes. They mean the
Pairing,
same.
in a way, yes, but when we say pairing of homologous chromosomes, we are moreover talking about it. If we want to see in detail, pairing of homologous chromosome is associated with synapse formation. However, the crossing over event is the characteristic of the pachytene, which will be ultimately lead to the chiasma formation. Okay, so pairing they are complete sister chromatid will come close proximity to the together, which is happening case of the A, and the crossing over event happen in the different stage of the prophase, but both are the part of the prophase one. Next.
Okay, ma'am. Ma'am, option C.
Okay, how about you, Danish?
1 second now.
Yeah.
Ma'am, I think anaphase 2 option D.
Anaphase 2, option D. Okay. So the question is, identify the meiotic stage in which the homologous chromosomes separate while the sister chromatids remain associated at their centromeres. So here we have to see the homologous chromosomes are separated. Homologous chromosome separation is characteristic of the meiosis 1. Okay. And sister chromatid separation is characteristic of the meiosis 2. So here we have been asked about the homologous chromosomes are separated, but sister chromatids are remain associated together. So sister chromatids are chromatids which are present in a single chromosomes, because each chromosome is made up of the two chromatids. The two chromatids which make the single chromosome is called the sister chromatids. So here we are talking about the separation of the homologous chromosomes, means each one of the set, means chromosome one set from the father and one set from the mother, which is overlapped, recombined, whatever it is, and it gets separated. So homologous chromosomes are, means their sister chromatids will remain the same. This is a characteristic of the anaphase 1, because in case of the anaphase 2, sister chromatids will be getting separated. On the other hand, in case of the anaphase 1, homologous chromosomes will be get separated, but their sister chromatids remain same. So option C will be the correct one.
Okay, ma'am, understood.
Last.
Ma'am, option B.
Yes.
Option B. Yes, option B is the correct one. Here, more than the words that they are using, just remember the concept what they are talking about. Here we are talking about anaphase. What happened in anaphase? Separation or segregation of the chromatids or chromosomes will be happening irrespective of that. So if something is affecting that particular process, the segregation won't happen.